talk to you about several things today. One is just the exam and some general test taking tips. And then also something that we didn't get to last session that I want to talk a little bit about at the end of the time today is the Hardy-Weinberg equilibrium and how to do those calculations. All right, so let's first of all talk about the test. And by the way, the test format that I'm talking about is for the in-person written version of the test. The digital version of the exam will be a little bit different. If you're taking that, make sure you look up some specifics about the digital version. I think most of you are taking the written test, so that's the one that I'm going to talk about today is the written version of the test. First of all, in that written exam, the first section of the test is multiple choice. You're going to have 60 questions, and you'll have 90 minutes to do that part of the test. That part of the test is 50% of your overall exam grade. Some of the questions will just be individual standalone questions. Some of them, there'll be 4 or 5 questions that'll be in a group, like on the ACT, where you'll have a paragraph to read, maybe a graph to look at, or a data table, maybe multiple items to look at along the way. Let's see. I see someone raise their hand. Just go ahead and unmute and ask your question. Looks like Skylar. Go ahead and ask your question, Skylar.
Yeah, I'm really sorry to interrupt, but
It's okay.
I just got my PC set up and I don't know what the Pear Deck code is. I'm sorry.
All right, that's okay. That's okay. It's TBJBHK.
Thank you.
Yes, sir. All right, so this part of the test, the multiple choice part, is the part where if you're going to run out of time, this is where it's going to happen. Like I said, this is very much like an ACT science kind of test. And if you've taken the ACT science section, you know that time is very valuable. So don't waste a lot of time. If you get hung up on a question, skip it. And then if you have time to come back at the end. This is the part of the test where you've got to be very aware of your time. You do not want to run out of time. The second part of the test is the free response. There are 6 questions. Again, you'll have 90 minutes. And this part of the test, again, is 50% of your overall test grade. I'm going to tell you the free response part of the test is where you end up earning your 3, 4, or 5. You've got to write. You've got to get some points on this part of the test. There are some very easy points that we'll talk about as we go through this that you should all be able to get. You've got to make sure that you get those points. The way the 6 questions are broken down, there are 2 long questions. Those are probably should take you maybe about a page of writing to answer, and a page apiece. And then there are 4 short questions that you can probably answer in 3 or 4 good solid sentences. You do want to write everything in complete sentences. You want to use ink. We'll talk about one exception to that in a minute, as far as graphing, but you want to use ink as much as possible. So the long questions are worth between 8 and 10 points. It can vary from question to question. And the short questions are worth 4 points apiece. The first long question, question number 1, is always going to be an interpret and evaluate experimental results question. So they're going to give you some data. They're going to give you probably a paragraph that talks about an experiment. And they're going to ask you to answer some questions about that experiment. Question 2 is going to be very similar, except this one is always going to require you to graph. You're going to have to construct a graph. Could be a line graph. It could be a bar chart or histogram. It could be a scatter plot. And very oftentimes these graphs are going to ask you to draw in error bars, or what we call 95% confidence intervals. And we'll talk more about those here in just a minute. The short answer questions, like I said, about a paragraph a piece is what it's going to take you to answer. The first one, again, is going to be sort of here's an experiment, analyze the experiment kind of thing. 2 is going to be more like a standard question. It's going to be a concept and answer some questions related to a concept. The third short question is going to be Answering questions about a model or some type of diagram or chart or visual representation. And the fourth one is going to be about analyzing some data. Again, very much about an experiment. So that's the general layout of the questions. Most people have plenty of time on the free response. Where time is of an essence is on that multiple choice part of the test. I will tell you though, make sure that, again, you're writing complete sentences. Make sure that you answer the question that's being asked. There is no need to give an introduction and a conclusion. Just answer the question. Get to it, get in, get out. Be concise, to the point. If it asks you to give 3 examples, then only give 3 examples, because the first 3 are the only 3 they're going to read. So don't give more than the question asks you to give. Answer the question, move on to the next part. That's important. And somehow I just messed up screen sharing, so let's see if I can fix that. All right, let me reshare my screen. I'm not sure what happened there. So let's try that again. All right, so you are going to have a formula sheet. If you're not familiar with the AP Biology formula sheet, make sure that your teacher gives you a copy. You need to be familiar with all the formulas on that sheet. You need to know how to use those formulas. You'll be able to use that formula sheet throughout both sections of the test, the multiple choice and the free response. You're also going to need a calculator. You probably won't need to use it a lot, but you can use the calculator throughout the whole test. And essentially, you can use any calculator as long as it can't access the internet. So that includes just basic old 4-function calculators, scientific calculators, and also fancy graphing calculators. You can use any of those as long as they can't get on the internet. That's the key thing. So let's talk a little bit more about graphing. When you're graphing, some really important points. First of all, your independent variable always is going to be plotted along your x-axis. And remember, your independent variable is the variable that you are intentionally manipulating during the experiment. You're changing it to see what effect it has on the other variable, the dependent variable. The dependent variable should always be plotted along the y-axis. Make sure that when you're doing your plots, that you clearly label each axis and that you include your units. Usually you put your units in parentheses, something like maybe meters per second or something like that. So have units on both axes. Make sure you clearly label. If you're asked to do 95% confidence intervals, plot your points, and then at each point, you're going to go up from each of those points. You're going to go up 2 standard errors of the mean. And those are going to be given to you. The SEMs are going to be given to you. You're going to go 2 times the standard error of the mean up. So let's say this is our point. And let's say our standard error of the mean is 2. So we would go from this point, we would go up 2 times that. So we would go up 4, and we would draw our first error bar. We would also go down 2 times the standard error of the mean. So in this case, down 4. Make sure that your error bars are connected to your point by nice vertical lines. That's an error bar. That's a 95% confidence interval. And essentially what that means is we can be, from this measurement, from this sample, we can be 95% sure that the population mean for this measurement lies somewhere in between the top and bottom error bars. So make sure you can do error bars. They're on every exam. You can do those on bar graphs. You can do them on line graphs. You can do them on scatter plots. Essentially, any kind of graph you're going to draw in this class, you can put error bars and create 95% confidence intervals on those. Here's the exception I talked about with pencil and pen earlier. I'm going to recommend that you do all your graphing in pencil. And then once you're finished and you know the graph is the way you want it to be, then color over it with a pen, because they want everything in pen. The reason you want to do it in pencil is your graph has to fit on the axis they provide. If any of it, even your error bars, go over the top or out the sides of the graph, you're not going to get credit for doing the graph. So plan it out. Make sure it fits on the axis that they provide for you. Always put your graph on that axis. And here's another important point. Sometimes the question will say, do a line graph. Sometimes it'll say, do a bar graph. Sometimes it'll just say, construct a graph. If you have doubts about the kind of graph that you need to create, just do a scatter plot. Because essentially the scatter plot can give you all the qualities of a line graph or a bar graph, and it'll count for both kinds. What a scatter plot looks like is this. Essentially, you just plot your points. Independent variable down here on the X, dependent variable over here on the Y, plot each of your points. And then notice that we've got error bars, up and down, 2 standard errors of the mean from each of those points. All right, another thing that I want to talk about the exam is what are called the command terms. Sometimes you'll hear these called task verbs. So these are the words that are almost always in bold in the free response questions. And again, they're pretty much always verbs. These are the terms that essentially tell you how you get points on the AP exam. And I've got a short list. I'm not going to go through every one of them, but I want to point out a couple of things about each one to you. If you're asked to calculate, they're looking for a numerical answer. If they give you a table to put values in, simply do the calculation, put the values in the table. That's probably all you need to do. If, on the other hand, they give you a lot of open space in the question, you need to show your work and then put the final answer in some kind of sentence form with units. So say the rate of doing cell respiration is blah, blah, blah milligrams per second. The next one I want to mention is if it asks you to construct, that's where they're going to want you to do a graph, maybe a table, and most of the time they want you to fill in the diagram or the table or the graph axis that they provide you on the paper. If they provide it, you need to use it. Describe is sort of a tricky word in AP biology. We all kind of know what describe means, but when they use this term in AP biology, this is really what they're wanting you to do. They're wanting you to explain how two things are related to each other, how they're connected. So specifically state both and how one affects the other. That's crucial. You've got to relate the two things in the question. If you're asked to draw or circle, make sure you do it on the picture or in the location where they tell you to do it. Almost always, again, they're going to give you a picture, they're going to give you a chart, they're going to give you a graph if they ask you to circle. Do the circling where they tell you to circle it. If they ask you to explain, you're going to make some kind of claim. So you're going to say, increased oxygen concentration increases the rate of cellular respiration. That's your claim. Then you're going to provide evidence. When you're providing evidence, use the data that they give you. So if they give you a chart or they give you a graph, you need to refer to that chart, to that graph, and then explain why you think what you're thinking. So make the claim, talk about the specific evidence that's provided in the prompt to you, and then explain why you think your claim is correct. That's important. Identify is the one word you might get on an AP question where a one-word answer will work. If they say identify an organism that can do both photosynthesis and cell respiration. You could say a plant. That works. I would still recommend you go ahead and write it in a complete sentence, but that's the one place where you might get by with a one-word answer. Maybe the favorite word of AP Biology FRQ question writers is justify. So when you're justifying, essentially you're providing evidence and explaining your reasoning. So you're going to essentially make a claim, and then you're going to refer to the data table, the figure, the diagram, the graph they provide you, and you're going to specifically talk about the type, the magnitude, the direction of the effects. So make sure that you, the key thing with justify is make a claim, provide evidence, refer to the evidence given to you in the question, and then explain why you think what you think. That's important. If they ask you to propose a hypothesis, not a null hypothesis, just a regular hypothesis, you might say something like increased CO2 concentration causes plants to perform photosynthesis faster, something like that. Simple case. Now, if they ask you to propose a null hypothesis, I want you to essentially say your null hypothesis for every situation they ask. The data that I observed or collected is not significantly different than the data that I expected. That is really all you need to do for a null hypothesis. There is no statistically significant difference between the data I observed or collected and the data that I expected. All right, they might ask you to propose a refinement. So here they're probably going to give you an experiment with issues like too many variables, or the control doesn't match up to the other experiment. So in that case, you're going to have to say what's wrong with the experiment and then how you could fix it, how you could make it better. So again, just a few command terms to pay attention to. Any questions on anything that I've talked about so far today? Anybody? All right, well, we're going to end today, the last 30, 40 minutes, talking a little bit about evolution and Hardy-Weinberg equilibrium. So first of all, make sure that you know this definition of evolution. Evolution is simply a change in the allele frequency of a population over time. Remember, an allele is a form of a gene. So essentially, evolution happens when a certain form of a gene becomes more common in the population across time. And note that populations evolve, individuals don't. You cannot evolve. Individuals don't evolve. The group evolves. Individuals do not. So we have some requirements for evolution to take place. The first one is that the population has to have genetic variability. If all the individuals in the population were genetically identical, evolution couldn't happen. There have to be genetic differences. And those can come from things like mutations. They can come from immigration, individuals moving in and bringing new alleles, or individuals leaving with certain alleles. Second, more offspring are produced than can survive. This is where natural selection comes into play. Nature kills. Some organisms survive, some don't. The organisms that are better adapted to that specific environment are the ones that are more likely to live. Now note, some adaptations are good in one environment, but not in another. It's not that one gene's better than another. It's that one gene or one trait is better in a specific environment than it is in another. But really the key to evolution is number 4. There's differential reproduction rates. The individuals that are better adapted to the environment reproduce more than the ones that aren't. And because those individuals reproduce more, their allele frequencies increase. The frequencies of the alleles they have, which means the frequencies of the alleles that everybody else has, those decrease. And that's really evolution in a nutshell. We talk about evolution being the survival of the fittest. Fitness in terms of evolution is not about how strong you are or how fast you are. When we talk about fitness in an evolutionary context, fitness is about reproductive ability. So individuals that are more fit are better at reproducing. They reproduce more. That's what fitness really means in an evolutionary perspective. All right, that being said, let's talk about Hardy-Weinberg equilibrium. So this is sometimes called genetic equilibrium. So we're going to use these two as synonyms. Hardy-Weinberg equilibrium is essentially a theoretical construct when evolution is not happening, which means allele frequencies are constant. Now, in the real world, that probably almost never happens because evolution is pretty much always happening. But essentially what Hardy and Weinberg were able to do is put a mathematical formulation on evolution. And also they could define what evolution is by defining when it's not happening. And essentially what they said is that there's 5 requirements for a population to be in genetic equilibrium, for evolution not to occur. And here are those 5 requirements. First of all, there can be no mutations. Because remember, a mutation is a new allele. And by definition, if you get new alleles, allele frequencies have changed. So number 1, no mutations, specifically those in the germ cells, because those are the mutations that end up in the sperm and eggs and are passed on to other individuals. All right, number 2, no immigration. And that's immigration coming in or immigration going out of the population. Because new individuals would bring new alleles, or individuals leaving the group would take alleles with them and change allele frequencies. Sometimes we refer to immigration as gene flow between populations. Next, the population needs to be large. And this is so that we don't get what's called genetic drift. Genetic drift is when essentially evolution happens by chance. It happens usually in small, isolated populations where allele frequencies just change by just random chance. In large populations, that's very unlikely to happen. Number 4, there can be no natural selection. Essentially, this means each individual has an equal chance of surviving. And then related, number 5, there can be no sexual selection. This means each individual has an equal chance of mating. Mating is random. Mates aren't being chosen. Mating is random. Every individual has a chance to mate. All right, and then down at the bottom, we see the mathematical equations for Hardy-Weinberg equilibrium. So let's start with this most basic one. P plus Q equals 1. So P is the frequency of the dominant allele. And that frequency is usually going to be given in a decimal, like 0.25. Q is the frequency of the recessive allele. Together, those two frequencies have to add up to be 100% or 1. So this equation is about allele frequencies. P and Q represent allele frequencies. Now, on the other hand, this equation is about genotype frequencies. Individuals that are P squared, that's the frequency of individuals that are homozygous dominant. So they have 2 dominant alleles for a trait. 2PQ, that's the frequency for individuals that are heterozygous for this trait. And Q squared, frequency of individuals that are homozygous recessive for the trait. Now, there's lots of different kinds of Hardy-Weinberg problems that you might be asked, but I have 3 of the most common types of problems you'll see on the AP exam on the next 3 slides. So we're going to work through those 3 problem sets. And if you have any questions along the way, stop me and ask me what's going on. So here's the most basic kind of problem. So in humans, brown eyes are dominant over blue eyes. And by the way, that's an oversimplification of the genetics. In a population of 1,000 individuals, 750 have brown eyes. Important term, assume the population is in Hardy-Weinberg equilibrium. Those equations, P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1, can only be used if the population is in Hardy-Weinberg equilibrium. So if it doesn't say this, there's another way we're going to work the problems. But since this prompt says population is in Hardy-Weinberg equilibrium, we're going to use these 2 equations. Notice the first thing it asks us for are the frequencies of the dominant and recessive alleles. Remember, the dominant allele is P, and the recessive allele frequency is Q. And I'm going to tell you that when you're working this type of problem, the thing you want to find first is Q squared. And that's the frequency of individuals that are homozygous recessive. Because remember, these individuals, even though their genotypes are different, their phenotypes are the same. These 2 look the same because they both have a dominant allele. This one has the recessive phenotype. So it's easier to separate from these other 2. So when we can find the homozygous recessive phenotype, or genotype rather, that's what we want to find first. So let's do that. So we've got 1,000 individuals, 750 have brown eyes. Well, brown's dominant. Well, if out of 1,000, 750 have brown eyes, that means 250 have blue eyes. And blue eyes is recessive. So the only way to have blue eyes is to be homozygous recessive. That means that 250 out of 1,000 is Q squared, the frequency of the homozygous recessive genotype. So if we divide that out, we get Q squared equals 0.25. Well, once you get Q squared, the frequency of the homozygous recessive genotype, you take the square root of both sides of that, and you end up with Q equals 0.5. That's the frequency of the recessive allele. So that's part of our answer to number 1. Q is 0.5. Now, remember this equation, P plus Q equals 1. So if Q is 0.5, that means P also has to be 0.5 because P and Q have to add up to be 1. So our answer to part A would be and I would write this out in a complete sentence, the frequency of the dominant allele is 0.5. The frequency of the recessive allele is also 0.5. Part B, how many individuals would you expect to be heterozygous? Remember that heterozygous is 2PQ. It's this part of the equation. So let's multiply 2 times P times Q. So 2 times P was 0.5. Q was also 0.5. So that turns out to be 0.5. So that means that 50% of our population, 0.5, should be heterozygous. Now that's not the answer. This asks how many individuals. This is the percentage or the frequency of the heterozygous genotype. 50% of all of our individuals are heterozygous. We had 1,000 individuals. So let's take 50%, 1,000 times 0.5. Our answer on part B, 500 individuals are heterozygous. All right, and then finally, what's the expected frequency of each possible genotype? Well, remember our genotypes are homozygous dominant, heterozygous, and homozygous recessive. And remember that in our Hardy-Weinberg equations, this is p squared. This one is 2pq, and this one is q squared. So let's just plug and chug and get an answer here. So our frequency of homozygous dominant individuals is p squared. So that's p was 0.5, so 0.5 squared, 0.25. We've already figured out our heterozygous frequency. It's 2 times p, 0.5, times q, 0.5. So 2pq is equal to 0.50. And then lastly, q squared. Q was 0.5, so let's square 0.5. And that comes out to be 0.25. So again, I would write this out in a sentence. And I would say the frequency of the homozygous dominant genotype is 0.25. The frequency of the heterozygous genotype is 0.5. And the frequency of the homozygous recessive genotype is 0.25. All right, that's it for the first problem set. Can I answer questions for you guys about that set of problems? All right, well, here's the second one. So notice on this one, out of a population of 60 people in a small town in Kentucky, one person has blue skin, an autosomal recessive trait. By the way, this is a real thing. 3 people in the population are carriers for the trait. Assume the population is not in Hardy-Weinberg equilibrium. And remember, that means we can't use the equations. We only use the equations when the population is in Hardy-Weinberg equilibrium. So on a problem like this one, we essentially have to count alleles. So we have 60 people. For most traits, 2 alleles, one from each parent, control that trait. So 60 people times 2 alleles apiece. That means there's 120 total alleles for this trait in this population. Out of that 120, this person has blue skin, which is autosomal recessive, which means this person has to have 2 recessive alleles. We'll just call those little r's right now. And these individuals that are carriers, they have to have 1 recessive apiece. So if you add those up, that's 5 total recessive alleles. 5 recessive alleles out of 120 total alleles gives us the frequency of the recessive allele, Q. So we could leave it as 5 over 120, or we could go ahead and divide it out. And that ends up being 0.042. So that's the first part of our answer. That's we're on B here. What are the allelic frequencies? So the frequency of the recessive allele is 0.042. Now, for the dominant allele, if 5 of the alleles are recessive, that means the rest of them are dominant. So that means there's 115 out of 120 that are dominant alleles, P. And we could leave, again, we could leave it as 115 over 120, or we could go ahead and divide it out. And when we do that, we end up with P equals 0.958. And that's the dominant allele frequency. So for part B, I would say the frequency of the recessive allele is 0.042. And the frequency of the dominant allele is 0.958. And on part A, how many total alleles are in the gene pool? Well, that's 120. We got that by taking the total number of individuals and multiplying that by 2. Because most of your traits are controlled by 2 alleles, one from your mom, one from your dad. All right, that's question set 2. Any questions on that one? All right, well, we've got one more question set to look at. Question set 3. So in Caucasians, hair straightness or curliness is governed by a single pair of alleles that shows partial dominance. Sometimes we might call that incomplete dominance. Individuals with straight hair are homozygous for the big H allele, while those with curly hair are homozygous for the little h allele. Individuals that are heterozygous, big H, little h, they have what's called a wavy hair. So it has some curls to it, but it's not all tight curls like a curly-haired individual would have. So in a population of 1,000 individuals, 322 have straight hair, 378 have curly, and 300 have wavy. And we want to calculate the allele frequencies for big H and little h. And notice it doesn't tell us that this population's in Hardy-Weinberg equilibrium. It asks us, is the population in Hardy-Weinberg equilibrium, which means we're going to have to do some calculations. So I've already done some allele counting on the next slide, so I want to show you how that works. So let's first of all calculate the frequency of the dominant big H allele. There were 322 people that had straight hair, big H, big H. So we take 322, multiply that by 2, because each of those individuals have 2 big H alleles. So that gives us 644 dominant big H alleles. There were also the people with wavy hair. There were 300 of those people. Those were the heterozygotes. Each one of those people had 1 H, big H apiece. So 300 times 1 gives us 300 more big H alleles in our population. We add these two together, and that gives us 944 total alleles. Well, since there were 1,000 people, that means there were 2,000 total alleles. So 944 out of that 2,000 total alleles gives us a big H allele frequency of 0.472. So that's our big H allele frequency, 0.472. Now we do the same kind of calculation for the little h allele. There were 378 people that had curly hair, and they're homozygous recessive. So if we multiply that 378 times 2 little h's apiece, that gives us 756. Remember, there were also 300 people that were heterozygotes, individuals with wavy hair. Multiply that by 1 little h apiece. If we add these two together, that tells us the total number of little h alleles. So that's 1,056. If we divide that by the total number of alleles, 2,000, that gives us a little h allele frequency of 0.528. All right, so if we go back to our question, we just answered part A. We calculated the allele frequencies, big H and little h. Now, to figure out if this population's in Hardy-Weinberg equilibrium or not, we're going to have to do a little bit more work. And the way we're going to do that is calculate some expected numbers and run a chi-square analysis on it. So these were the observed numbers: 322 straight hair, 378 curly, 300 wavy. So our expected numbers, we would find those by using the Hardy-Weinberg equations. And remember that these individuals, straight-haired individuals, these would be the p squared people. These would be the q squared because the curly is recessive. And this would be the heterozygous condition. So to calculate these expected numbers, we would take the frequency of big H, the dominant allele, and square it. So big H frequency was 0.472. So p squared would be 0.472 squared. And then we're going to multiply that by the size of the population, which was 1,000, to get an expected number. So we would expect there to be 222.78 straight-haired people if this population was in Hardy-Weinberg equilibrium. Do the same kind of calculation for the curly hairs. So Q squared, the frequency of the little h allele, the recessive allele, was 0.528. So we're going to square 0.528. That gives us Q squared of 0.279. And then we multiply that, and actually I need to carry that out maybe a couple more places, 278.78. Multiply that by 1,000 to get the number of individuals. Get 278.78. Repeat that calculation for the wavy-haired individuals. So 2 times P, 0.472, times Q, 0.528. And when we do that, that gives us 2PQ is equal to 0.49843. Multiply that by 1,000 to get the number of individuals in the population, and that gives us this number. All right, so these were the numbers that we actually had. These were the numbers that we expected if the population was in Hardy-Weinberg equilibrium. So now we're going to compare these two sets of numbers using a chi-square analysis. And we're just going to fill in that table to do that. So O minus E. So in this first row, it would be 322 minus 222.78 to get this number. 378 minus 278.78 to get this one. And 300 minus 498.43 to get this one. Now we're going to take this row and square it to get this row. So 99.22 squared to get this, 99.22 squared to get that, and negative 198.43 squared to get this. Finally, we need to go O minus E squared over E. So that's going to be this row divided by this row. So 9,844.61 divided by 222.78 to give us this. And then repeat that all the way down. Remember, to calculate a chi-square value, the formula for chi-square is the summation of O minus E squared over E. That means to get our chi-square value, we need to add up this column of numbers. Well, I've already done that on the next slide. So I'm just adding up 44.19, 35.31, and 79.00. And when I do that, I get a chi-square value of 158.5. Before I go any further, though, I need to think about a null hypothesis. My null hypothesis is that there's no statistically significant difference between the data I observed and the data I expected. So I've got a chi-square value. I now need to compare that to the critical value. This has to come out of the chi-square table, which I don't have in front of me right now. But we would have, in this particular problem, we would have 3 different phenotypes. So we would have 2 degrees of freedom. It's always 1 less than our number of phenotypes. And then you always look at the 0.05 probability row, and you get that out of your table. The critical value in this case is 5.99. We compare the chi-square value to the critical value. If chi-square is greater than your critical value, which it is in this case, we reject the null hypothesis. Remember, our null hypothesis is that there was no difference. Well, we're rejecting that, which means there is a difference between this set of numbers and this set of numbers. And since there's a difference, that means our population is not in Hardy-Weinberg equilibrium. Now, on the other hand, if chi-square had been less than the critical value, that would mean that you fail to reject the null hypothesis. And in that case, that would mean that our 2 sets of data are maybe a little different, but the differences are small and due to chance. And that would indicate that the population was in Hardy-Weinberg equilibrium. But in our actual problem, our chi-square value was much greater than our critical value. That means we reject the null. And that tells us that because those two sets of data are so different, the population is not in Hardy-Weinberg equilibrium. All right, that's it for my presentation today. Couple of quick reminders. I'm going to tell you that the two most important units on the AP exam are Unit 7. That's the evolution unit. And Unit 6. And that's the unit that's about like gene regulation. So if I were you and I were studying for this exam and I had a limited amount of time, I would go to AP Classroom and I would watch the videos from these two units, the evolution unit, Unit 7, and also the Unit 6 gene regulation videos. Also remember that on the A+ College Ready YouTube channel, it's A+ College Ready Science. We have a playlist of AP Biology review videos, and they cover everything in the course. So I would recommend you look at those. Also, there's an AP YouTube channel, and they've been running AP exam review videos on that YouTube channel for about the past 3 or 4 weeks. They're all posted on their channel. So I would, again, before I got ready for the exam, I would go to AP Classroom and watch all the Unit 7 and Unit 6 AP Classroom videos. I would go to the A+ College Ready Science YouTube channel and find things there that you're not familiar with and watch those videos. And I would also go to the AP YouTube channel and watch as many of those videos as you get a chance to do. All right, again, last minute things, multiple choice, be aware of your time. Make sure that you know how to use all the formulas on your formula sheet, and you should know how to do the calculations and what the calculations mean. When you're doing the FRQs, don't get discouraged. There's going to be questions you don't know how to do. That's the way it's supposed to be. But there's also going to be really easy parts to most every question. You've got to answer those kinds of questions. You've got to get those. Also, make sure that you do your graphs correctly. Make sure that your graph completely fits on the axis. Make sure that your independent variable is on the x-axis. Your dependent variable is on the y. Make sure that you label those axes. Make sure that you include units. And when you're asked to do error bars or 95% confidence intervals, remember it's 2 standard errors of the mean up from the point and 2 standard errors of the mean down from the point. Final thing, if you don't know which kind of graph to make, just plot your points. Do a scatter plot. And go ahead and put the error bars on that scatter plot. All right, are there any questions you guys have for me before we go? All right, your exam is on the 14th. If you're taking the regular first administration, it'll take you about 3.5 hours or so to do. Again, take your time, read those questions carefully, answer the question that's asked. I would read each question twice just to make sure that I'm answering the question that's asked. Don't be intimidated by the vocabulary. There's going to be a lot of that there. Read the question, get to the point, answer it. Again, I want to thank you guys for coming to all these sessions throughout the year, and best of luck to you. I hope you all make 5s.